Thanks for tuning in today. I'm Dr. Nick, the Incrementalist. Join me as I seek out the small incremental changes being applied in other industries that we can learn from and that can be applied in healthcare. Can these changes bring immediate value, but also add up to the big improvements and revolution we need in healthcare? Come along with me to explore the possibilities. My innovative guests from around the globe have used small incremental improvements to achieve their moonshot.
And today I'm delighted to be joined by Dr. James Liu. He is the president and co-founder of Helix. James, thanks for joining me today.
I appreciate it, Nick. Thanks for having me. It's really exciting to be here on Incremental Healthcare.
So, as I do with all my guests, I think it's important to understand people's background. Like so many folks that I have the privilege of interviewing, you've got a fascinating background. You're doubly qualified. Tell us a little bit about your journey and how you got here to sort of share your experiences.
Yeah, I'm happy to. I would love to say that it was pre-planned, but certainly was not. I think it only makes sense in retrospectively. But prior to helping to start the company at Helix, and that was started in 2015, I had just joined the faculty at Duke University, and I was starting a laboratory focused on machine learning methods, or now called kind of AI type methods on medical records, as well as translational genomics. And I was supposed to finish my clinical residency and my clinical training there, really focused on internal medicine, and then ultimately into kind of crit care and pulmonary. I guess I'm proud to say I'm a residency dropout. So I'm no longer qualified actually to see patients, but I think the medical training and obviously the research components have been a big contributor to what we're doing here at Helix. Prior to that, trained as an MD PhD. Most of my PhD work was mostly on what was then the very early days of genomics. So I remember I worked with Dr. Richard Gibbs, who's my primary advisor, and he runs the Human Genome Sequencing Center at Baylor College of Medicine. That was one of the few places in the United States doing very large scale sequencing back when sequencing was really brand new. So that laboratory sequenced Jim Watson. I think at the time he was the second person in the world being sequenced, and I worked on some of the early projects on the first tens and hundreds of people being sequenced. And a long time ago, I spent a little time in venture capital, a little bit of time in pharmaceuticals. And a long, long time ago, I trained as a chemical engineer at Stanford. So that was my origination of my career, but kind of had this interesting trajectory kind of throughout. Very focused on large scale computational ideas. How do we leverage data to make it useful, and how do we think about application of new technologies to healthcare?
So, there's a bunch of things in there that are really interesting, not least of all the sort of early phase when sequencing, although, it dates back to the first individual and, the big genome project. And I forget what the numbers are, but, if it's not billions, it's certainly a lot to sort of sequence that one individual. And then we've seen this, incredible decline. As you were sequencing the hundreds, thousands of patients, had the cost fallen sufficiently, or was it still at the point where it hadn't really dropped to sort of expand the access?
Yeah, it was kind of in this intermediate point. So just as a reference point, back when the Human Genome Sequencing Project was done and completed, and the reference was done, I think, in 2003, I think that project cost something on the order of $3 billion. And today we sequence routinely, I think, around $600 for a genome, at least on the research basis. And so if you look at the curve, it looks like Moore's on Moore's law. And just for the audience, Moore's law is the doubling of transistors per chip every 18 to 24 months. And so you have this halving of cost effectively for performance. We had sequencing had a curve that was exponential on top of an exponential. And so the costs have dropped dramatically. But I remember, back in the day when we were doing the first pediatric rare disease exomes, you were begging and borrowing just to sequence the parents. Because that really helps you adjudicate the case, understand the pediatric case. But sequencing an additional individual costs tens of thousands of dollars at that point. So not billions of dollars, but tens of thousands.
Right.
And so it was very hard to do. Now we obviously routinely sequence in the thousands of people per day. Certainly not tens of thousands across the country. So we've come a long way. But I do think from that cost component is just one piece of it. I think that the knowledge component of it increases exponentially as well as we reduce costs. And I think that's been a big driver for increasing utility there.
So we see this, exponential decline. I think the projections based on that curve is that it will very soon be about the price of a toilet flush, which opens up new opportunities in that space. Tell us a little bit about Helix and the formation of that company. What was the genesis of that, and how did you arrive at the idea?
Yeah, I mean, the ideas really started percolating in the early 2010s. And, at the time, the world was sequencing what was then the first kind of 1,000 people. And I think the central insight really was that, in the world of healthcare, there probably is only one data set that doesn't change with time. And that actually is your germline genome. And that's the genome that you inherit. And, uniquely, if you can generate the data at very high quality, and then leverage it in the healthcare system on a digital basis, you can come back to that over and over again through your life. And it can inform different parts of your care as you kind of progress. And so, Helix is formulated on that idea, what we call sequence once, query often. But there are two main components to what we offer. The first one is, can we generate high enough quality data to make sure that we can use the data over and over again? And the second part is, what does the data platforms look like to enable the healthcare system to use that data over and over again? And I would say that the first part of that data generation is a problem that's gotten easier with time, particularly as technology has gotten better. But the part about orchestrating use of data is something that I think continues to be a major challenge, certainly for people who are working within the American healthcare system. And so we spend a lot of time focused on that data platform and utilization of that data platform.
So there's a number of things sort of buried in there. And the first one that sort of jumps out at me, and I think it's important to explore for those listening, is, you talk about the data set that's not going to change. A couple of things in there. One, do you think that's ever going to be different? Obviously with gene editing and, put the ethics to one side, because I know huge challenges, but I mean, I think conceptually it's possible. And then the other is, if that's an unchanging model, I think most people, those that are old enough, will, snap back to Gattaca and think, well, wait a second, are you saying I'm pre-programmed to end up at this, there is no change because this is my genome makeup? I think there's good reasons not to believe that, but I think it's important to explore. And tell us a little bit about your thinking around that.
Yeah, happy to. I mean, maybe I just make a very clear stance, which is that the genetic code is not deterministic. Mm-hmm. And so this idea that you're pre-programmed, I think, is, for lack of a better word, just false. Certainly, there are pieces of the genome which we know on disease do influence the likelihood, and in some cases, very strong likelihood of disease manifestation. But most things in the genome are probabilistic, and they interact with the environment, and they help, to some extent, I think people describe it as setting the stage. And all your interactions through the rest of the world and all these other, what we consider other omic technologies that do have a time component of them, all come into play, and they influence both the day-to-day experience as well as the either diseases or the behaviors that you exhibit over time. And so we certainly view the genetic component as something that will be informative for various pieces of your care over time. But we definitely don't think that for most of this, that this is something that's set in stone when you're born.
So, I mean, I think entirely correct. I think people sort of struggle with that because, there's some element, well, that's my programming and, you don't end up. But back to the sort of point of, changing the code, what about diseases? I mean, we have diseases that, we can point to a single point of genetic change that you could, now you've got to get it early enough. So there's all sorts of chat. And I know I'm way out there, but it's not often I get people with this level of insight. You think there's potential for that to come about, putting the ethics to one side and the challenge that surrounds it? Yeah,
I mean, I would say just on gene editing, and maybe I can give a little context, right? What we've gotten very good at is obviously reading the genome. Mm-hmm. And the second piece is like writing the genome, which I would consider gene editing. And that technology has progressed very rapidly in the last couple of years, starting with something called CRISPR-Cas9.
Right.
And I think there's, Jennifer Duda and that team kind of won the Nobel Prize last year. I think it was last year on this one. The technology there is incredible, and there is the potential that you could be thinking about ways to edit the genome in ways to eliminate these very point mutations that result in very deleterious diseases. But I do think we have to be very careful about where we apply that technology and make sure that technology is applied to the right set of problems. We're often, I think, what often gets dreamed about, which is like, oh, we're going to edit all sorts of places in the genome, do all sorts of stuff, kind of more on a hypothetical science fiction type basis. It feels a little bit like technology searching for a problem versus the problem getting solved. And so what I don't want to do, and I don't think that anyone in the field wants to do, is introduce secondary issues. Right. As well as the technology is not perfect, right? The fidelity is not 100%. And so you do introduce other errors potentially through the writing process. And so I don't think that's something that we understand quite well enough that we would think about it from a general basis. But I think there are very specific problems around some of the germline diseases that can be worth having this discussion around.
Fantastic. I mean, I think, exciting, but obviously we have to sort of think very carefully about how we progress. Lots of challenges, and I don't want to sort of diminish any of that. And I'm not sort of advocating for one way or another, to be clear. As I say, not often I get people that really have deep insights to be able to ask those questions. And I know, certainly one that comes up a lot in the conversation. So
sure.
Back to Helix and, what the purpose is of that organization. You sort of teased a little bit about the challenge of gathering that data, the challenge of sort of making it useful, the fidelity or the accuracy. Tell us a little bit about the platform and what you've done and what you've managed to achieve so far.
Yeah, happy to. I think one, maybe I'll talk about the two components. One is the data generation side, and one is the kind of data platform side, and how they kind of intermingle and some of the things we've achieved there. I think one is, starting on the data generation side, we've been very focused on making sure the data can be used over and over again and generating the platform for that. And so one of the things that we were quite proud of earlier this year is to achieve the first FDA clearance for an exome platform. And I think that was fairly unique in that if you read the documents carefully, what they've done is essentially a few things that we've actually advocated for. The first one is the FDA has accepted the concept that you can decouple generation from usage. So you can generate a data set in the past at high enough quality, and then you can use it for an application set in the future that may not have been contemplated at the time you generate the data. So that decoupling, I think, is fairly unique in the molecular space because most of the time you generate a data set, you use it immediately, and you don't come back to it. So I think that's first. The second one is this idea of digital use, right? So we're going to re-query the data, develop a new application or a current application, launch them as we go, and making kind of a digital resource and building a regulatory pathway around that one. And so I think those concepts conceptually and then together really enable a world where we're thinking about the usage of those data over time for each individual and generate a regulatory quality that you can accept for a wide range of regulated applications in healthcare. On the usage side, beyond the regulatory side, a lot of it is around how do you manage the physician workflow, particularly as we imagine this technology being used by frontline physicians. So genomics, I think rightfully, like most technology, starts in the specialty space. The specialty space is a bunch of power users who have specific workflows and smaller patient populations. And I think we've achieved incredible things in the oncology space, some of the rare disease spaces, pediatrics, et cetera. Now, when you move out to primary care and you're using it as a primary genetic screen or to identify people at risk for disease, your frontline physician now needs different sets of tools to be successful. And so we're very focused on how do you kind of work on that interaction? How do you educate physicians and patients to do that? How do you make sure, frankly, you get the outcomes that you want? Because much of this is new in many ways. And I think this is a problem that not only will impact genomics, but also do you think about other molecular, other omic technologies or other data technologies coming down the road? How do you build a bridge between the frontline physician, the patient, and the healthcare system to enable them to adopt new technologies that we know will improve patient care, but doing it in a way that's efficient, doesn't take 30 years of new medical training. And so our ability to develop new technology and deploy certainly developing technology is certainly much faster than our ability to deploy and adopt. And so how do we kind of accelerate the adoption curve, I think, is something we're very focused on.
So for those of you just joining, I'm Dr. Nick, the Incrementalist, and today I'm talking to Dr. James Liu. He is the president and co-founder of Helix. We were just talking about the platform, the exciting FDA clearance. I mean, that's not an easy journey. There's certainly been some stumbles and falls along the way. I'm sure that didn't help you, or maybe it did. Maybe there were some insights from that. I'm curious to understand, was there some elements in that past that you look back and think, well, from an insight perspective, to get it through that process and start to deliver value, were there specific things that you brought to the table or that you changed or adapted to that really changed the trajectory for Helix and, obviously this FDA clearance? Yeah,
I mean, I think much of the things that we've been saying for this idea around digitizing the genome, I think, has been an idea that we started propagating in 2015. I think it's taken, frankly, it's just taken time to educate the various constituencies and stakeholders around how do you even think about a digital resource over time. And I think to some extent, the macro trend has been that healthcare is becoming increasingly digitized. And those what many people call the exhaust of the healthcare system. So the claims data, the laboratory data, et cetera, are actually resources that can be used to improve the care of patients and either in the day-to-day services component of it or even for things like drug discovery. And so we've been writing a little bit on that macro trend, but I think we've been very focused on the genomic aspect of it specifically because we do think that one is unique in that aspect. And so part of it's been the education of it. Part of it is, frankly, building the proof points. And so, for example, one of the big things that we've been working on demonstrating is that as we go wide, certainly for everyday access, one of the key components and key questions has been, does it make sense to screen every person in the United States genomically? And for what conditions and what things are impactful? And so we had this very nice paper that we put out in Nature Medicine, I think it was mid last year, actually, in the middle of COVID, that we wrote with one of our big health system partners, Renown Health, on a population of about 30,000 people in northern Nevada, where we screened every single person unbiased who walked into the program, looked at the incidence of genetic disease across just 3 conditions. We looked at whether or not they would have been ascertained in the normal healthcare system, and then what the disease outcomes look like for these patients. And just to ask some very fundamental questions of, does this impact clinical outcomes? And what we found is something like 1 in 75 individuals are carriers of a very deleterious genetic marker for one of these 3 conditions. And the conditions are Lynch syndrome, which is a syndrome of cancer, hereditary breast and ovarian cancer caused by BRCA1, BRCA2, and familial hypocholesterolemia, which is inherited high cholesterol. So just, 9 genes, 3 conditions, 1 in 75 people carried. We found about 80% or 90% would not have been ascertained in the medical system at all. So they either did not have family history or were not asked family history or would not have been discovered until disease manifestation. But we know that these patients can be monitored and intervened early on to prevent. And so I think it was a pretty big finding that it was, greater than, it's like 1 in 1.33%, but that 90%, 80, 90% level tells us that the healthcare system is not equipped to detect these individuals. And so genomic screening becomes basically probably the one and only tool to really identify early risk. And shouldn't we be doing something to identify? And I think in the system, at least in the US, typically patient populations that are highly morbid, that can be detected with technology, we should go find those patients and do something about them. And so even on a very narrow basis, our belief now is that it has very clear benefits from a clinical perspective. We think it has clear economic benefits for the healthcare systems writ large. And this is something we should be deploying. And so we're seeing it actually in all of our programs now. Renown is doing it universally. Mayo Clinic, at least in our program with Tapestry at 100,000 patients, is also doing what we call a CDC Tier 1. So this set of diseases, screening universally for that program. We're starting to move that to other health systems now. And I think we're at the very early stage of saying, look, it's going to be a general standard of care benefit over time. And then we'll be able to leverage those data over and over again in a wide variety of different cases.
So important to understand, and we'll put a link in the show notes to the paper, but what's the sensitivity and specificity around those? You talked about the 9 genes and the 3 diseases.
Yep.
And specifically, I'm focusing on the false positives. Does that even occur? I mean, is that a problem? How did that show up in the study?
Yeah, so the sensitivity, like if you look at all the potential carriers of those genetic carriers of markers, I think we're probably around 90 to 95% sensitive. I think there is probably a small set. If you expand the gene list a little bit or expand the way you look at it, you can probably expand slightly. The specificity, which is, I think, a really interesting point here, and it's super important because you don't want to ascertain a bunch of people who actually won't manifest disease. So we have very stringent criteria about which genetic mutations or variants are likely to be protein disruptive and then would have manifested in disease. And so we think the specificity is quite high. And when we look at the disease population, so we just took an all comers. If you look at all people who had carried and you look at just, incidence of disease, what we found is something like 26, 27% of them had already manifested disease, which is, 10 times relative to the normal population distribution. And so if you just took that snapshot in time, you actually do find that there's an incredible amount of disease burden in that population. And the question then for the rest of the population is, would they manifest with more time? And what we see is, yeah, we strongly believe that would happen. And so when you draw these curves, it looks like something like 50 to 60, 70% of people as they get older will manifest some level of disease because of genetic.
Interesting. So, huge opportunity. And I think, that list is obviously going to expand as you start to understand more. In the few minutes remaining, tell us a little bit about COVID-19, because obviously, there's a element there, and that's, ever present. What's been going on? What's been your experience?
Happy to. I mean, in the last year, Helix has become one of the largest diagnostic labs for COVID-19. But I think most recently, actually, we've been a big partner and contributor with Illumina and CDC on surveilling the country. And I think one of the key things we've been very focused on is variants of concern for early detection, frankly, for not only this pandemic, but we think about infrastructure for the next pandemic. So we've been building some of those data and sequencing capabilities right up our alley, kind of the sequencing side, on making sure that we are ready for that, not only for the continuation and hopefully end of this pandemic, but also for anything in the future. And so we've been one of the key organization identification of the what's B117, which is, probably unfortunately named the UK variant.
Right.
And, certainly we made predictions in, I think, February this time frame, and it was just published in Cell, that this would be dominant in the US by around early April. And that's what we're finding now. That unfortunately, this recent uptick in cases here in the U.S. is likely due to both more contagious strains coupled with reducing restrictions on individuals from a social distancing perspective, as well as increased travel. And so, if anything, right, increasing surveillance allows us to get a picture of whether or not we're going to see, frankly, continuation or longer tail, essentially, on what's happening here in the U.S. So we continue to test regularly. We're doing, we're operating 24/7 a day on the diagnostic side, and certainly on the surveillance side, our goal is to continue to sample as deeply as possible to really detect any of these variants of concern.
Wow, that's I think hindsight's always 20/20, but not surprising when we look at the data today to sort of think about B117 and the increased transmissibility, and even there's increases in morbidity that we've seen as well. So concerning, but the ability to sort of predict that at the point when you were back then, that's great news. We just have to sort of enable more of that. My sense is we're going to see more of this from, companies like yourself, capabilities like yourself, because we've learned so much more. Closing minute, where do you see this going? What's next?
Just on the COVID side specifically, I do think there's an incredible opportunity being generated right now around surveillance and the new vaccine technology. And the idea that you could surveil a new variant, understand its phenotype, so its clinical course, understand if it escapes vaccine, and then get a new vaccine into market in kind of the months time frame, I think is not only transformative for COVID, but for all future sets of infectious diseases. And so it's been, obviously it's been a tragedy the last 18 months, but I think the deployment of U.S. and certainly worldwide expertise in life sciences has been incredible. And watching it in action and moving so quickly, I think has been tremendously rewarding. And certainly for me, incredibly optimistic about where we're headed. So I think the deployment of new sequencing technology and coupled with these new life sciences technology, I think we're going to be in a much better place here in the next couple of years.
Couldn't agree with you more. I think, it's terrible to talk about silver linings with such a tragic sort of experience, but that's exactly what I get out of all of this, and I agree 100%. Unfortunately, as usual, we've run out of time, so it just remains for me to thank you for joining on the show. Thanks very much.
Thank you, Nick, and I really appreciate the time and the interest.
Thanks for joining me today. Do you have any better ideas, or have you found a small incremental change that's brought about a big improvement in your world? Let's continue the conversation on our hashtag, The Incrementalist, or share with me at DrNick1 on Twitter. You can find more information about the show on our program page at healthcarenowradio.com. And tune in next time to hear my discussions with leaders and innovators from around the globe who've revolutionized their space by using small incremental improvements to achieve their moonshot. I'm Dr. Nick, the Incrementalist, and I'm starting a revolution through evolution.